All right, hello and welcome to our first Physics and Astronomy lecture series lecture presentation of the year. My name is Dr. Aaron Quinter. I'm the Professor of Physics and Engineering Program Coordinator here at SWOC, and I'm happy to have all of you joining us virtually for today's talk. We are privileged today to have a guest in Dr. Jason Hafner from Rice University. He is a professor of physics and chemistry at Rice University in Houston. He had studied at Trinity University to get his bachelor's in physics in 1993. He earned his PhD from Rice in 1998, working under Dr. Richard Smalley, working with the carbon, the carbon nanotubes and the carbon-60 buckyballs, if I remember correctly. He joined the fleet in 2001. Dr. Hafner currently does research looking at nanotechnology and looking at applications sort of gold nanorod plasmons, and looking at information, or looking at how to get information and how to do applications thereof. So rather than me try to explain it, I will turn it over to Dr. Hafner. Please feel free to ask questions in the chat. If you're watching, we will save those and we will ask those at the end as part of a Q&A. And so I'll turn it over to Jason.
All right. Well, thanks, Dr. Corner, for the introduction. It's great to be here with everyone tonight. And we're going to, like, as he said, we're going to talk about biomedical applications of nanoscience. And let's just take it away. Yes. So actually, this title is my more technical title. And it is what we're going to talk about, lipid membrane structural analysis by something called enhanced Raman scattering. So we'll get to the details of what that means, but I was going to say some general things about nanoscience just to give you the big picture, so to speak. So let's go ahead and jump on to slide 2 here. Let's see. So nanomaterials looks like, yeah, I'm a little wide. Nanomaterials are interesting because what you can do is you can take a normal material that you think about, and when you cut it down to small nanometer scale size, you get new properties. That's sort of the big picture theme in nanoscience and how things got started. So I've got a few pictures here of just some examples, and these are really sort of the classics. You know, if you went and studied a recent literature on nanoscience, it would be just an infinite number of new materials. So I'm only telling you the really old, introductory ones and how things got started. So one is carbon. So we know carbon has two forms. Oh, good, there we go. Two forms of carbon macroscopic. There's a graphite form that makes, natural graphite that you might find, or in a pencil lead, which is not lead anymore, it's graphite. And of course, diamond is the other form. But graphite is made up of these little flat sheets of carbon. And if you make those into a sphere, you have the famous buckyball. I was too late to work with Smalley on buckyballs, but I did find them lying around the lab. So the research was pretty much over. But if you fold them into a tube, it makes a carbon nanotube. So these were a big subject to study in the 90s and getting nanoscience started. So as you can see on the top, you have your macroscopic graphitic carbon. And on the bottom, that's sort of a vertical layer of nanotubes that they're pulling to the side to make fibers. And these are very strong, interesting, electronically interesting fibers. In more modern times, there's something called graphene, which is currently all the rage. And that's just sort of take your sheet of single atomic layer of graphite and don't roll it into a tube. Just leave it flat. And then the detailed structure and the edge structure of that sheet will give you interesting electronic properties and other properties. So one is just folding carbon into weird shapes. Another one is semiconductors. Of course, in the bulk, we often think of semiconductors as being wafers. We make wafers, we make computer chips on them, etc. So there's a picture of a person holding two pretty large semiconductor wafers, probably silicon. If you break those silicon pieces down to the nanometer scale, a few nanometers across, and you put them in solution, and you shine blue or UV light on them, they fluoresce, and they fluoresce all different colors. So it's something the bulk doesn't do, but the little pieces will do. And in this case, it's a sort of a quantum size effect. You can tune the color with the size of the teeny piece of semiconductor. So that's another example, making it small. And then there's metals. So we all know how important metals are, largely due to their high conductivity, and in a few cases due to their value, like the gold bricks I'm showing you there. But if you take that gold and shrink it down to really small sizes, put it in a solution, it also becomes a new color. And that color is actually tunable with the size of the particle, kind of like the semiconductor, but for a physically very different reason. So we're going to talk a little more detail about why gold does that. So a different reason, but the story is the same. As you take a bulk material, break it into small pieces, it has new things it can do. All right, so let's hit the next slide. So I could go through a comprehensive list of all the areas of science and technology and nanoscience, has impacted, is working on impacting, but that would be an hour talk in itself. The field sort of started with these interesting materials and got into applications, and then it grew and it kind of became everything. So it started, you could on a crass level say this is related to funding, or you could also say these small materials sort of interact with everything, so we can take it into many branches of science. Look at it either way you want to look at it. So what we're going to do tonight is just talk about light and some of the optical properties of one of the nanomaterials and what my lab is trying to do with it. So let's talk a little bit about light. So you probably know light is a wave, and it is a wave of the electromagnetic field, which is a combination of the electric field oscillating and the magnetic field oscillating. It's actually all one big field. And the light waves we think about, the ones that are visible to the eye, have a wavelength of about 500 nanometers. So, not really it's kind of big for the nanometer scale. But so let's think about two things, is that things can be affected by light. Okay, so, and here I wrote macroscopic light. By then I meant the wavelength is really long. So you shine, visible light on an atom, something really small. It can be excited by that light. It doesn't matter that the light has a much longer wavelength than the size of the atom. And the same thing for nanomaterials. They can be affected by this long wavelength light, and the wavelength isn't an issue. But if you want to sort of work with light at the nanometer scale, that's different. Then you need the light to become nano, So that's the second question. Can you manipulate light with nanomaterials, not just have them respond to it? So can you focus it? Can you transmit it? Can you change it? Can you intensify it? Can you radiate it? All these things. So we're going to talk about which nanomaterial will do those things, not just experience the light, but actually change the light. So let's hit the next slide. And the material we're going to talk about is gold. So here is the old interest in gold. Of course, finding gold is of significant interest. So there's some gold miners there panning for gold in a river. The scientific interest actually predates that 1800s kind of mining interest. So the fact that gold makes has an interesting nano-optic property goes all the way back to here, the Lycurgus cup from 300 AD in Rome. And of course, they didn't know what was going on. They didn't have electron microscopes, but they found that if they made glass out of the sand in certain rivers where they would tend to find gold, I suppose, it had this interesting optical property. So this cup is an example. If you put a light inside the cup and shine it, then the cup looks red. And if you shine light from the outside, the cup looks green. So the color depends on how you illuminate the glass, or in this case, how you illuminate the cup. So this was just, interesting, 300 AD. It was actually Michael Faraday who was able to show the reason, not necessarily physically the reason, but experimentally to figure out what was happening to the gold to make it do this. So he's figured out that it basically, when you go to really small pieces of gold, it does this. So not all the way to gold ion, but much smaller than just a bulk piece of gold. So I'm not sure if he actually was able to estimate the size, but at the time, colloid was the word for nanoparticle. So if you could get them down to sort of the nanometer scale size, suddenly the gold turned red instead of yellow, kind of like that cup when you looked at light on transmission. And then, of course, this was studied further. Actually, if you really want to get into the weeds here, there's something called Mie scattering, which was figured out by Mie, and M-I-E, not me personally, by Gustav Mie. And he actually, one of the main motivators of him developing Mie theory, which is the detailed scattering of light, is to understand this phenomenon. Why is it that small gold particles suddenly look red? If you look in the original paper, there's a resonance and it says, in German, gold nanoparticle and whatever. So it was a big mystery at the time, and they figured out it's basically just light scattering off of small gold particles. So you can understand it with just classical 1900 E&M. I have a third picture here just for fun. That's Zygmundi was an expert in microscopy, and here he's showing early dark field microscopy images of gold. And my picture, there I am being in the way, but those vials have gold particles of different size, and it goes from deep red to kind of yellow looking. But in the microscope, he sees them green. So the solution is deep red, but then he's seeing them as green. So that's a little bit of further mystery we'll be talking about what's going on. Thanks for getting me out of the way there. Okay, so we can jump to the next one. So what's going on is something called a localized surface plasmon resonance. So this actually is not a fancy, hard to understand quantum property. It's actually a classical, simple to understand property. It's just that it only occurs noticeably when the particles are at the nanometer scale. So it's not quantum, but it still requires things to be small. So really all that's happening is, if we think about a little gold particle like that yellow circle there is your gold nanosphere, and the sinusoid is the electric wave, the electric field going by. And what makes a metal is that the charges in the metal are free to move around. That's why it can conduct electricity. So an electric field pushes a free charge. Like the force on an electric field, F equals QE. So as the light goes by the sphere, it pushes charge up. It pushes negative charges against the electric field. And when the negative free charges move down, that leaves the positive atoms uncovered. So you essentially make a little electric dipole in the particle. So you have excess electrons on the bottom, that's negative, and the top is positive. And then then the light wave keeps going and it flips direction. So it's basically the light, which is an oscillating electric field, drives the charge to move up and down in a sinusoidal pattern in time. So it's just shaking the electrons is all it's doing. But if you think about it, if the electrons try to leave the gold, they're going to be pulled back, because even though the field, even though the light is pushing them up, if you leave a positively charged nuclei and try to leave, it's going to suck the electrons back. So they're being driven out, but there's a restoring force pulling them back in. So ultimately, it's like a mass on a spring. The electrons are just oscillating up and down. It's like a mass on a spring. It's like a driven mass on a spring even. So you could just tweak the electrons and they might shake and damp out, but the electric field is actually driving them. So like this mass hanging on a spring there, drawn underneath, you may have done this in a lab at some point. If you drive a mass on a spring, or maybe you have a flexible beam that acts like a mass on a spring. If you drive it at different frequencies, so eventually you'll hit a resonance. So if you drive it near its natural frequency, you'll get a strong resonance as you go through. And that's really exactly what is happening with the gold particles, and that's why they look red. The resonance is actually in the green. So there you can see a very pure solution of gold particles is the little red vial. And finally, to the right is a spectrum, an extinction spectrum of that solution of gold particles. And you can see very clearly that very strong peak. That's the resonance. The spectrometer applies all different wavelengths of light, and then just measures where is the absorption the strongest. So basically, the wavelength from 400 nanometers to 1,000, that's sort of visible in the near IR. That's basically tuning this oscillation of the electrons, and they oscillate really big at about 520 nanometers, and that's where the resonance occurs. So 520 nanometers is actually green. So if you shine light on these and look at it scattering back, they look green, but the scattering is actually pretty weak. The other way to see the color is to have a solution of them, like that red bottle, and have white light coming through it. And then what happens is it takes the green away. So if you have white light, you take the green away, it basically looks red. So this is why the Lycurgus cup sometimes look green and sometimes look red. When you put the light inside, the resonances took the green away and you saw red. When you shine light from the outside, you see green. It's often hard to see the green because these are, if you do really small particles, they don't scatter much. So the green scattering is not as easy to see. But that is the fundamental effect that the gold nanoparticles of various sizes and shapes have with light. But so far, we haven't told you that they manipulate light. That's still, in a way, that's just kind of like experiencing light and absorbing it. So in the next slide, we can go and go to the next slide. Let's see what is happening near the particle. So it turns out when these electrons slosh up and down in the gold, they create a very strong electric field. So basically, normally you have your gold there, it's full of electrons, it's full of positive nuclei, everything's neutral, the electric field is just zero. But imagine you take a bunch of the free electrons and you shove them to one end of the gold and put them in a little tight spot. What you're going to do is create a strong electric field there. Think of it as like Coulomb's law. You know, you have a field created by point charges. So if you have an unbalanced charge on the top, you'll get a big field there. And if you leave positive unbalanced charge on the bottom, that'll make a big electric field as well. So, and because this particle is small, it sort of makes that electric field in a very small region, nanometer scale. So it's not driven by the size of the wavelength of the light, it's driven by the size of the particle. So that's what these graphs are here. So if we look at A, this is what happens if you send the light at a single spherical particle. And this top bar, the color graph, is E over E naught squared. That basically means the electric field you get over the original electric field. So dark blue is kind of the strength of the electric field of the light. And then the color bar is how much bigger the electric field is near the surface of the particle. So the little white arrow on A, that's telling you which way the electric field was pointing as it went by the sphere. The sphere isn't shown, it's just where there's the dark blue circle. So you can see at the surface of the sphere, there's a region on either side where the electric field points that gets an enhanced electric field. And so the enhancement, you look at it, okay, it's like 10. Okay, that may or may not mean anything to us that it's 10, but the key is the spatial size. So if you look, that's from -15 to 15 on the horizontal axis, and that's in nanometers. So you can see all that enhancement is sort of in a scale of only about 5 nanometers, very sharply focused. So this is a case where the nanomaterial, the gold sphere, has manipulated the light. It's actually focused it down to that little region. Now it's still light, right? So you might say, I've heard of this thing called the diffraction limit, and you can't do things smaller than the diffraction limit. So that's true for just a plane wave. So this is no longer a plane wave. This is a plane wave that has been focused to the nanometer scale. It doesn't mean you can immediately use it to do high resolution microscopy. It doesn't mean these are X-rays, because it doesn't focus it into a form of light that you can just propagate across the room. It focuses it into a form of light that is bound to the surface of the nanoparticle. And it actually isn't really propagating at all. It's just a light that's kind of shaking by the surface of the nanoparticle. So it's a very unusual kind of light. But the key is it has focused it to the surface. Now that enhancement is sort of 10, according to the color scale. Yellow is kind of like 10. But then you can start playing games at the nanoscale. You can have a lot more than just a sphere. So the middle panel, B, is 2 spheres. Send the light in, polarize the same way, the electric field horizontal, and basically you get 2 for 1, right? So each particle is doing pretty much what the single particle did. It's just that there's 2 of them. But then you turn the electric field the other way. So in graph C, you can see the arrow is pointing up and down. And now you're driving the charge all the way across both particles. And that has a completely different effect. Then you get a case where there's positive here, negative there, positive there, complicated. We don't need to go through the details. But you can get a case where you get a huge electromagnetic field enhancement in that gap. So now you can kind of barely see the shadow of the 2 particles. But inside there's this gap, and you see the enhancement is now 750. So that's extremely intense light in that little teeny volume. So, I'm citing one paper here, a very good paper where I got this figure from, but this was studied like very thoroughly in the 2000s. As you can imagine, people studying all the different geometries and also spectral features. You know, all of these peaks don't necessarily happen at the same place the individual nanoparticles peak is. So how the peak shifts, how to manipulate the enhancement was a huge field. It still is, but it was widely studied in the 2000s. This is just sort of the simple example of one sphere and two spheres. But so this is the key, the beginning of the key of how we're going to use this to start to study biomembrane structure, is that we can now get the light focused down to a few nanometer scale. All right, so let's look at the next slide. So you're going to see LSPR. So when you see that, just remember you don't need to remember exactly what it stands for, Localized Surface Plasmon Resonance, but that's basically this effect when the free electrons oscillate on resonance and create these strong electric fields. So also in the 2000s, another big thing that blew up was making all these different shapes. So through chemistry, we gained the ability to make different geometrical structures of gold particles. And it turns out there are LSPRs you could change. You could tune them around in the visible. You can make them broad. You can make them narrow. And one key thing is you can tune them into the near infrared. So this was, and still is, a big part of nanomedicine. And a lot of it was by my colleague, Naomi Hollis here at Rice. And she made one of the very first tunable particles, and it's the one on the left, the nanoshell. So a solid sphere, the resonance is at about 520, like I showed you. But if you take a hollow sphere, or you take a piece of glass, like a dielectric sphere, and you coat it with gold, then looking at the ratio of the radius of the glass sphere to the gold thickness of the gold shell, you can tune, and you can tune the peak and the near IR. And that's interesting biomedically because tissue is relatively transparent in the near IR. So you could put these particles into the body and then shine light on them and see where they went, or even heat them up to burn and burn up a tumor under the skin, etc. So a lot of effort has gone to that. And it's actually in human trials, and it's actually it's being used. I believe it has actually made it into the clinic. So that was really a big deal. Around 98 is when it first got going, because that was the first one. And right on its heels, or essentially at the same time, gold nanorods were synthesized in high yield and high quantity. And that second one, the rods, you can tune them through the visible and near IR just by changing the aspect ratio. So just like taking that sphere and elongate it, and that makes the peak shift. And then a plethora of structures have come out. That cage is like a hollow cage. We discovered these nanostars. They have a resonance for every tip. All kinds of cool stuff showed up. It was a big fad to name them after plants and food. So people found all these structures, and it was always like, some sort of flower or like a hamburger or something. So nanostar was as far as I went in terms of these weird nanoparticle names, but it got entertaining for a while. We were predicting what food would show up in next week's copy of Nanomedicine. Okay, so I just want you to be aware there's a lot of structures. So we're going to actually look at nanorods later. And there's just one more slide about all the things that you can do with these is, gold particles scatter very brightly. So they might be a complementary thing to use relative to fluorophores. So, fluorescent molecules are widely used for molecular imaging and biosciences. So there you can see that's a image of a chromosome that's stained as it normally would be non-specifically with fluorophore. And they stained it with silver particles to bind at a specific gene on the chromosome. And that's what that blue is. The blue is scattering from silver particles. They can scatter in the blue. And it's just showing that these particles can be just as bright, as fluorophores. My lab for a while did work on bioassays. So basically you can put antibodies, that's the panel on the upper right. You can put antibodies on these particles and then flow and try to catch the antigen. And then the binding of the antigen, the thing you're looking for, will change the plasmon resonance in a way you can detect. So we worked on that, showed some fundamental things about the interface, but then I'm not much of a technology guy, so I didn't really pursue it. And of course, now I'm kicking myself because everybody needs an assay now. But no, that's the other people, smarter people are working on that and working on assays. A third one is the one I already mentioned, that you can use this to bind the particles to certain cells specifically, and then use a laser to just blast those cells. That's been done in vitro and cell culture a lot, but it is making good progress in tissues and in real, human use in the clinic. RLACE, I think, was the name for a while. So look into that. I hope we can all benefit from that someday. Okay, so now that was sort of the big picture intro to nanoscience and a few examples of its use in biology. So now the next slide is good. So now I'm going to tell you about, so I used to do these bioassays and what everybody is interested in and what I could actually get funding to do. And then I decided, this is really successful and everything, but I don't want to be successful. I want to do what I want to do. So what I'm going to tell you about now is a weird, crazy thing that I'm doing that I don't recommend anyone pursue. But basically, we're just working on to see if we can use these in a new way to get biomolecular structure. So if you've taken any biology class that talked about molecules at all, it probably mentioned the importance of the structure-function relationship. Our basic understanding of how proteins work, the first thing you want to know is how are they put together. So all these beautiful images in your biochemistry textbook of protein structures, that's because that's what you need to know. And they're even doing it in sort of an omics level at this stage, finding different types of structures that seem to always go together. And that's what this sort of 4D or whatever this plot is. It started categorizing proteins by their structure because structure is so important. Structure tells you function. So a huge amount of effort in the sort of molecular biology world is structural biology. So a lot of effort at just getting these structures. And how do you do it? Well, the most powerful way is you get the protein, you make a really pure form of the protein, and you get it to crystallize. You make a crystal of protein, just like you make a crystal of salt or a crystal of some simple inorganic material as a crystal growing kit that you did when you were a kid. You can do that with an entire protein. It's a little bit harder, but a lot of effort goes into crystallizing them. Once they're crystallized, you send X-rays through them, and then the diffraction pattern of the X-rays can pretty much directly give you the protein structure. Don't ask too many details. It's not quite that clean of a analysis, but basically the diffraction gives you the protein structure. The other big one is NMR, nuclear magnetic resonance. And this tells you, the angles between bonds and the structure, and then they can put a lot of that together and figure out, not quite at the powerful level of a giant macromolecule like X-ray can do, but NMR gives you a lot of structural information. Those are the big two, and then there's many others. Structural biology is a very big field, and not something I would go in and say, I have a new way to give you protein structure, because everybody knows how to get protein structure. So we're focusing on something else, and that's the next slide. So so biomembrane structure. So lipid membranes, these are, of course, very important. This is the boundary of the cell, and it's also the boundary of almost all the organelles. People that study membranes say that everything is membranes, and not quite true, but a lot of stuff happens in the membrane. So it's important we understand it, and unfortunately, it's very hard to study, because the reason the membrane is a membrane is because its components are amphiphilic, which means half of them love water and half of them hate water. So the fundamental component is the little phospholipids, which are just fat molecules, basically, and they have a head group that likes water, and they have these carbon chain tails, the fatty acid acyl chains that hate water, and you throw them in water, and they make this sheet like you see there. And they just do it, they just self-assembly. They just do it naturally. It doesn't take a lot of work to get them in this form. So you say, okay, I could maybe study those, but then in the membrane are millions of different kinds of molecules. There's sterols, there's membrane proteins, then there's like lipids with other things stuck on them, and it's all just a big crazy mess. So it's not really a system that you can study structurally. Like you can't crystallize it. You have to crystallize an entire cell, get a bunch of identical cells and crystallize the whole cell, right? You can't do that. So basically it's just too big, and it has too many components, and it's too dynamic. So therefore it resists a lot of the standard methods of structural biology. And really the most fundamental one is the bottom right one, is they're not even soluble in water. These molecules, the part that hates water makes it insoluble. So if you put it in water, it immediately starts to form membranes. It's not like you can sort of isolate them and treat them as individual entities in water. So that's what makes membrane structure very difficult. And that's why a lot of physicists that get into biophysics end up studying biomembrane structure, because that's where all the mysteries are, and that's where the need for new tools is large. A lot of that is in biomembrane structure. So let's look at an optical tool that we could use for biomembrane structure in this extensive introduction. Okay, so you know about nanomaterials, you know about membranes, and now let's talk about something called Raman scattering. Okay, so we talked a little bit about light, right? Light is the oscillating electric field. You shine light on a dielectric sphere, and or a metal sphere, any sphere, and the electrons oscillate up and down, and then they radiate light to the side. So that's kind of the cartoon you're seeing here. That beam is a laser beam going to the right. The gray circle is like a dielectric sphere or a gold sphere, what do you want to call it? Just a particle that's going to scatter the light. And it scatters the light away. And what is the color or frequency of the scattered light? No, it's the same as the laser. Why would it be different? So the laser causes the electrons to jiggle at the frequency of the laser. That jiggling radiates light, so it's the same frequency. So that plot is like as though we had taken a frequency spectrum of the scattered light. And you can see it's just a peak at omega naught, which I'm saying was the frequency of the laser. So that's pretty boring. So what would happen if we replaced that sort of spherical cow sphere there with a molecule? So that's the next slide. We'll turn that into a molecule. I was trying to minimize my animations. So if you put a molecule there, something real, then what happens is suddenly you see extra peaks. You see the big strong peak at omega naught. Most of the light is at the original frequency, but then you get peaks on either side of that peak. And those are what was discovered by Mr. Raman. And what's happening is the light is interacting with the vibrations of the molecule. So really light deep down is a photon. Light is a particle. So when it scatters off of the molecule, it can pick up energy from a vibration, and then the photon is a higher energy, which means it has a higher frequency, which actually means it's to the right in that plot. Or the photon can come in and dump some of its energy into a vibration and then come out at lower energy. So you can actually use the visible light scattering as a way to get the vibrational spectrum of the molecule. And vibrational spectra are a very powerful way to analyze molecules, to figure out what they look like and what's happening with the molecule. So it's discovered, I forgot the year, 20 something like that. And if you read the original paper, it's pretty fascinating. There was no spectrum to record because that equipment didn't exist. I mean, photographic plates, I think, existed, but his original paper was just by looking at it with his eye. Like he saw the bands and described them, and it's just text, right? This is a very early photographic plate showing you some of the Raman lines. Anyway, so let's look at how we're going to use Raman scattering for membranes. The next slide. All right, so the Raman scattering gives you the vibrational spectrum, and sort of vibrational spectra, it's what sometimes they call the fingerprint spectroscopy, because as you can imagine, big complicated biomolecules like that lipid structure. So the thing I'm drawing there at the bottom, the chemical structure, that's the fundamental unit of a membrane. That is a phospholipid, that's phosphatidylcholine. So the right side with all the phosphate and the nitrogen and everything, that's the hydrophilic side that likes water. You can see it's all charged. And the left side is the carbon chain that hates water. You can imagine if you were to think about how could that vibrate, it can vibrate a lot of ways. You could take every atom, it has 3 degrees of freedom. And I think you can get you get like hundreds of ways this thing can vibrate, hundreds of normal modes, as we call them in vibrational analysis. So for small molecules, you can look at the spectrum and you can know what that molecule is because it has a few vibration peaks that you can identify. When you get into big molecules, it kind of gets to be a mess. So if you look at those spectra on the right, the top one is that molecule, phosphatidylcholine. The other two are just different lipids. And you can kind of tell that's got a lot of peaks in it. And see, the ones with numbers are the ones that they're pretty sure they know what they are. We'll talk about how well they know in a little bit. But each one of those peaks with a number, they kind of know what it is. So what you can start to figure out is things about how the molecule is arranged based on where those peaks are and how strong they are. But it's an imprecise science, I would say. It's tricky. Like the peak at 1442, the one by the 3, that's due to anywhere that there is a CH2 or a CH3 vibrating a certain way. But there's CH2 and CH3 all over the molecule. So there aren't actually that many peaks that are just localized to a certain place. But if you can get those, you can learn things about the structure of the membrane. A lot of the things that are learned are not like from first principles. I know this vibration, I know exactly what's happening. It's more phenomenological. You know, we look at these peaks when the lipids are in this phase, and then we look at them when the lipids are in this phase. And we start to figure out, well, maybe it shifts that way because this molecular part is hydrogen bonded to that part. And in this phase, it's not. So it's a lot of indirect work that's been done over many decades to get a lot of powerful insight into structure from these kind of spectra. It's like great detective work. If you're into that kind of a thing, you want to get into vibrational spectroscopy. Unfortunately, though, we've looked at ourselves here. We're going down the rabbit hole. Unfortunately, it doesn't just spit out the full 3D structure like X-ray diffraction. It's sort of indirect structural analysis. Okay, so let's see, next slide. Okay, so now you have all the tools you need to understand surface-enhanced Raman scattering. And now we're to the thing that I actually use, okay? So surface-enhanced Raman scattering. So Raman, as I said, has been around for 100 years or so, Raman scattering. And one problem is it's very weak. You need a lot of molecules to be able to get a detectable Raman signal. It's not as much of a problem as it used to be. Now with powerful lasers and good detectors, it's pretty easy to see. But in the early 70s, people were looking at the Raman spectra of molecules while they were on an electrode, because they want to study like electrochemical effects of what happens to this electrochemical electrode when I put different molecules on the surface. And they were getting these freakishly large signals. Right? So the Raman spectra of pyridine absorbed at a silver electrode. They didn't put it in the title, but the point of the paper was it's much stronger than it should be. And so these are just three of the really early papers. The third one there, anomalously intense Raman spectra of pyridine on a silver electrode. So it took a while to figure out what was going on, but after a decade or so, they figured out that it's related to those plasmons I told you about. So we looked at the plasmons of a nice gold sphere, but even just a rough gold or silver film has these excitations and has these little regions of strong electromagnetic field. And if you sort of look through and go through the numbers, you find that those intense focused fields, due to the gold and silver surface, will enhance your Raman signal even more intense. You take the enhancement and you multiply it to the 4th power, and that's how much it enhances the Raman signal. So they were actually getting these strong signals from just small numbers of molecules on the gold. So this was discovered in the early 70s, intensely studied just to figure out what's going on. You know, what could this possibly be? In the 80s, they figured out it was electromagnetic enhancement. And in the 90s, people studied it. And then in the 2000s, they realized this would be a great platform for detecting small molecules, maybe explosive molecules or pathogens. So then there was a huge interest in finding substrates that would let you detect small amounts of molecules with this method. And I'm sorry, I meant to switch. That's what the next slide is kind of about, is that these large enhancements can be as high as 10 to the 8. So it depends really on how you make the enhancement. So there on the left, just a particle like we're working with, like a nanorod or a sphere, at the end, where you shine the light and it makes that little enhanced region, that's like 10 to the 5. But then you start making gaps. If you start making really small gaps, you can get 10 to the 8, 10 to the 9. People have reported 10 to the 14, I think, but that's probably not real. But you can get the enhancement so high, you can see the vibrational spectrum of a single molecule. And you can just sit there and watch it, and you can see as the molecule shows up and disappears and shifts position, you can see the spectrum change. So you are without a doubt watching single molecule vibrational spectra. So this is very powerful. So this is why there is interest in using this as a sensing platform. So if you've heard about SERS, or if you mention SERS to a scientist and they roll their eyes, that's because this was massively studied as a sensing platform. And it hasn't quite become a routinely used sensing platform because it's very sensitive to the chemical interface, which is hard to control. There's great progress, and I'm sure SERS will be in wide use very soon. But as a technology platform, it's very powerful, but still a little bit under development. And a lot of the work is finding how can we make a cheap substrate that gives us very high sensitivity, that's very robust, and that we know what we're getting. Because you can say, well, the vibrational information will tell you how you get your specificity, what molecule you're looking for. But if you're talking about big molecules, it's hard to untangle those vibrations. Like I mentioned with the lipids, you just see this big mess of peaks, and it's hard to know, dynamite, it's hard to tell. Okay, so the next slide then is what we're doing with SERS. So we've made it to my research. So we're almost 2/3 done, and we're finally to what I'm doing. So what we're doing, let's put it all together, is we're taking gold nanorods. So that's what this is, a TEM image very close of a gold nanorod, and you're only seeing half of it. So the dark part is the gold structure, and it's got a hemispherical end cap. We're shining light on it, And that's what the red and yellow is, like the enhancement region, enhanced down to just a few nanometer region. And we have ways to put a lipid bilayer on the surface of the rod. So I can give a whole talk proving that we've done that, but I'm not going to do that. You're just going to trust me that we have that little thing is like the top and the bottom layer of lipids, very small, around the surface of the nanorod. And this is in solution, so we can do this in solution. So really the rods are bouncing around, lipids are bouncing around in solution. They're coming on the rod and going off the rod. It's a very dynamic system. It's all very well mixed. But essentially, this is what we have, is a rod with a lipid bilayer around it. And we take that surface-enhanced Raman spectrum, and we get a bunch of peaks. And here, if you can get rid of my head again real quick, because there's a critical peak that I'm... Oh, wait, I can actually point to my slides. So anyway, so if you look here, CTAB, that was the surfactant. I'll leave me off for the rest of the slide, thanks. So the CTAB was a surfactant that was on the rods in the first place that's similar, actually, to the lipid. And then we switch it to DOPC, which is the lipid. So one great thing about SERS is we can tell we definitely switched it, because this is the peak that goes with the surfactant head group, this nitrogen vibrating around the nitrogen, and this is the lipid. So we know we completely switched to lipid. And then we can see other interesting things like this peak. This is this double bond stretching, vibration around that double bond. So you don't really see it there in the CTAB, there's some noise there, but you get a strong peak that shows up, a stronger peak that shows up with the lipids. And then these are all well-known peaks of the carbon chain in here, but the CTAB also has a carbon chain, right? So some of those also show up there. But this is just the main thing here. This is my evidence that we are definitely exchanging the surfactant CTAB, and we definitely have lipids. This looks exactly like a lipid spectrum on the surface of the rod. So now, so what? Right, so we can look at this and we could say things like, well, there's the head group peak, and there is the double bond. We could say, why is the head group peak stronger than the double bond? Because if you just do like a normal Raman measurement, it's the other way around. The double bond might be stronger than the head group. We could say, well, maybe it's because the head group is right up next to the gold, right? The enhancement is strongest near the surface and it decays away. So it's looking at that is kind of what gave us the idea of maybe we could get structure out of this SERS spectrum if we could really understand all the peaks. So now the next slide is our structural method. Okay, so now we're going to go through a little bit of like the math and the concepts of how we think about this. Okay, so we call this structural analysis by enhanced Raman scattering. So that's, you have to do these, abbreviations in science to get anywhere. So that spells SABERS. We needed a fancy name, so we called it SABERS. I kind of wanted to call it hierarchical arrangements from near field enhanced Raman, but my students shot that down. So we went with the SABERS. So this is really just sort of an analysis method, a way to use this data to get structural information out. It's just sort of a procedure. But the idea is this. So here we have sort of a spherical cow physics molecule. Okay, so the gray is just the cloudy, it's just the molecule. The green and blue are specific parts of the molecule that are vibrating. And then the spectrum shows you, say we have a peak that we know is due to the green vibration, and another peak is due to the blue part vibrating. So in principle, the relative heights of those two peaks in that spectrum could tell you which one is closer to the surface, but it could also tell you how they're oriented with respect to the surface. Because what drives the signal is not just how big the field enhancement is, but also how the vibration is aligned with the electric field. And the electric field is always normal to the surface. So we have a case where since it's SERS, since it's happening on the surface of a rod, we know the field is normal. So as the molecule turns, the signal will go up and down. And we actually get more information about the orientation than we do from the field changing with distance. The change with distance looks really dramatic, but actually at the scale of a molecule, it doesn't drop off that fast. The field strength at the front and the back of the molecule is different by 10% or something. But as you rotate through 90 degrees, the signal can go from 0 to full signal. So actually orientation is what gets us more information, as you'll see when we look at this. But you can't just interpret a SERS spectrum this easily. The heights of the peaks, there's many things going on. So there's just a list of all my grievances here. First of all, it's hard to even know what vibration each peak is. And to do this quantitatively, you don't know how many molecules are at the edge of the nanorod. If you're going to interpret the height of the peak, you got to know how many molecules you have. You don't really know how many molecules you have. The height of the peak is also affected by the spectral efficiency of your detector and your grading and all this other stuff and your optics. And then you need to know the field decay. And then fundamentally, we don't know how strong each mode is. Like in theory, how strong should this mode be? That's not really written down anywhere. So you don't really even know that. And then finally, you also have a problem that in addition to the SERS signal, you get a Raman at the same time of the molecules that aren't on the surface of the rod. And you would think those aren't enhanced, so it must be almost nothing, but there's more of them than there are of the ones on the rod. So it basically, we spent a decade getting through all these difficulties to where we could actually do some analysis. So the next slide is going to have the description, the mathematical description of the approach. So we don't need to worry about this in great detail. Oh, I'm sorry, this one's animated. If you could click, I thought I took the animations out. See, if you click, yeah, okay, click, click, click until I say stop. Oh my God, okay, keep going, keep clicking. Sorry, I meant to take all this out. Okay, stop there. Okay, that's good. Oh, go back one. There we go, good. Okay, so big ugly expression, yeah, go back one. Big ugly expression of all these things. We are going to talk about obviously how this was derived, but I just want you to realize that there's a lot. So this is how much Raman signal you get if you shine light with intensity I naught. So it depends on like how many molecules there are. This Raman tensor is basically describing, you know, what does the vibration look like and how does it couple to the light. So for Raman, it doesn't care about the orientation. For SERS, it does. So here, this is a combination of a bunch of tensor elements that's invariant with orientation. This is a single tensor element that says just the Z component. Right? So that's the difference between Raman and SERS. Here, it's all orientations. Here, it's just along the E field. Here's the enhancement factors that we added to turn this into SERS, etc. So all we do really is we take ratios. So now this next slide, I'll give you an idea of how we do this, not the exact details. Yeah, so here what we do is we say, okay, we got to get rid of all these unknowns. The number of molecules we don't know, the spectral efficiencies. So we take ratios. So here, for example, is a molecule. This is the surfactant, CTAB, that's originally on the rods. Here's the Raman spectrum of CTAB, and here is the SERS spectrum of CTAB. So clearly these two modes are showing up in each case. So the first thing we do is we take each peak and we take a ratio of its measured SERS intensity to its Raman intensity. So there's a number we can measure, right? The height of that peak and the height of that peak. And if you look at our expressions, that ratio cancels a lot of stuff out, right? That's the power of a ratiomatic method. You cancel things that you don't want. And when you cancel it out, you get an expression like this. It still has the number of molecules in each measurement in it, has the electric field enhancement factors and the tensor parts. So the problem is we don't know those big N's. We don't know the number of molecules in the beam spot and the number of molecules on the particle. But then we do another ratio. We take the little r ratio for 1, the little r ratio for 2, and we take a ratio of that. We call that big R, the big ratio. So the big ratio between peaks 1 and 2 is the little ratios, and basically that gets rid of the N's. Right? Now we don't have to worry about the number of molecules because we're ratioing two peaks in the same spectrum, and of course they're coming from the same number of molecules. So that's how we're using ratios to get rid of unknowns. We're left with a number that we can measure experimentally from those two spectra, and a number we can get theoretically in principle that depends on how far you are from the surface, that would be in the E's, and how you're oriented with respect to the surface, that would be in all these tensors. And we have these two numbers, and we can actually measure it, and then we can calculate it for all different orientations, and then compare. And that's what this crazy plot on the right is. So you can get rid of my face again real quick. Oh, no, you did. Is this color plot is basically a measure. So if you look at the scale between 0 and 1, when it's 1, you have a good match between experiment and theory. That means that the spectrum agrees with that orientation. And when it's 0 blue, you don't. So what this is telling, and then the two axes, one is the polar angle. The vertical axis is the polar angle. The horizontal axis is the roll angle. So what this map is telling us is we get a good match when the molecule is tilted at about 27 degrees. And it doesn't really care how it's rolling, okay? So what this says is basically the surfactant is sitting tilted at 27 degrees. So this was our very first structure that we figured out. So it's not dropping a 3D structure like X-ray diffraction, but it's giving us direct structural information, and we get it by taking the ratio of all these different properties. So that's how SABERS works, and that's on the simplest system of the surfactant. And we'll go on to lipids. But I'll tell you a couple of quick stories about getting there. So the next slide is how we calculate the fields. And we're going to skip that because it's really boring. There's a picture of the full field around the nanorod. The next slide, though, I think is important to point out so you can understand a little bit about our vibrational spectroscopy. So it's funny that when you read papers about vibrational spectroscopy, they always use the phrase, we assigned the peaks. And that's the definite euphemism. That is, they use the word assigned for a reason, is because you don't really know what all the peaks are. For like benzene, you know what all the peaks are, right? But for like a lipid or something big, there's a lot of guesswork and intellectually driven, figuring out what all the modes are. You can do lots of tricks. You can deuterate and do a shift in the peak due to the change in the weight of the hydrogen to the deuterium and the carbon-13. A huge amount of effort. I'm not saying it's all just guesswork, but I'm saying that the word assign is there because it's hard to know them. And because they want to calculate them. Well, how? Right? So it's really hard. It's a quantum system. It's a big molecule. So a way to calculate them has come along. In the early 2000s, there's a method called density functional theory that lets you calculate the electronic state of a whole molecule. Very powerful theory used massively in chemistry and physics. But the ability to do Raman is relatively new, early 2000s. And one of the early papers, I love what they say here, because you get into a field and you don't know the things that people don't say. The people don't write but say. It's hard to know those things. Every now and then somebody with a big enough name will write it down. And he did it. And here it is. In general, vibrational spectra of large molecules with many degrees of freedom are too complicated to yield information about the vibrational motions in detail. Experience and chemical intuition are necessary in order to assign peaks in spectra to particular modes of vibration. In most cases, this is not possible at all. Right, so this is the theorist throwing down the gauntlet and saying, you guys don't know what these peaks are. Here's a way to calculate them. And what is great is now 20 years later, you can buy packages that do these calculations. So big, dumb experimentalists like me can run the software. So I don't have to be an expert in DFT. I kind of have to know what's going on. But I can actually buy the software. I can run it on big molecules. The company says, those are too big. Please don't. I do it anyway. And actually, I get really reasonable results. So we get the Raman tensor I mentioned by running these calculations. So the next slide, I'll show you an example of, again, CTAB. We really haven't done lipids. I keep showing CTAB results. Here is the Raman spectrum of CTAB with several peaks. And we definitely know what this one is. That's the head group. And some of these we kind of know because it's an alkane chain. But this is how powerful that DFT method is. This is the match for a single CTAB molecule. But that's with a straight chain. So that's a pretty good match. That peak is covered, that peak is there, that peak is there. This region looks kind of similar. But we're getting even more realistic as they don't have straight chains. They have flexible chains. So they're called conformers. Every single bond can rotate, right? So we do a bunch of conformers and average them, and it gets even more realistic because that's actually what's happening in solution. That matches, that matches. We've now sort of built this match better than it was by looking at conformers. And the head group actually got messed up a little bit for complicated reasons. So a big part of this method is the fact that we can calculate extremely accurate Raman spectra of big molecules. And this is just not real well known in the field how accurate it is. People know, yeah, DFT can do that, but I don't think people appreciate how close you can get. And we're actually able to learn, forget about SERS, just doing this, we can get new insights into these Raman modes of lipids that people have thought about for like 50 years or so. So anyway, what I'm going to show you now is our SABERS results on a few lipid systems, and that's it. And then we'll conclude. So I told you the CTAB Bilayer we did. I sort of showed that in the how SABERS works part, and I guess I'm kind of repeating it here. But in the end, we got 27 degrees for the tilt of that layer. And if you look at what other people think, they get about 27 degrees. So the next slide is just some other work on the CTAB layer by more traditional methods, small angle X-ray scattering and small angle neutron scattering. And they actually found it was a little thinner than they expected. So they said maybe instead of 40 angstroms, it's 32 angstroms. Maybe it's interdigitated. But our result actually is very consistent with that. We say, no, it's actually just tilted. The reason, this method will just measure the thickness. So if you look at that thickness, it's really just that it's tilted. So our very first structure was in good agreement with other measurements, and actually, in my opinion, clarified them a little bit. The next slide is the phospholipid bilayer. So now we're actually doing the lipids, and now is when it gets complicated. But we looked at the lipid molecule as two molecules because we can't actually do the DFT on a full lipid, it's too big. So here's the head group vibration. That's this peak here, the one peak. And then this other little molecule is the double bond, and that's the two peak. And then all the junk in the middle. The red is SERS, the blue is Raman, the black spectra are the theoretical. But when we do this analysis here, so the multicolor graph, it's got the polar angle and the roll angle. You can see it's red and equal to 1, where the polar angle is 0. that tells us the lipids sit normal to the surface, which of course we know that's what they do in a bilayer. We can also further confirm it by looking at the matches that we moved the double bond away and closer, and we got the similarity really peaked right when it was 13 angstroms between the carbon-carbon double bond and the nitrogen, which is also correct for the bilayer structure. So our method basically, I wouldn't say we've discovered the lipid bilayer structure, it was well known, but our method is completely consistent with the lipid bilayer structure. If you look at the next slide, is just sort of a molecular dynamics simulation of a bilayer, just if you wanted to compare in detail. We don't have time now, but if you wanted to compare in detail, you could see, yes, the carbon-carbon double bond is exactly 13 angstroms from the nitrogen group. So we definitely got that spacing correct based on where it's widely accepted to be. The next one is the first time I would say we got new information that there was no other way to measure. And that is that using this method, we looked at tryptophan in a phospholipid bilayer. So tryptophan is an amino acid. It's one of the 20 that are on, the peptide chain that makes proteins. And it's kind of amphiphilic, right? It has a part on it that likes to the oily lipid environment. So it's often used to anchor membrane proteins. So there's a lot of interest in how does tryptophan just sit in a bilayer? Let's not put it on a protein yet. Let's do something simple. Just throw it in a bilayer and see what happens. And there's no good way to measure that. You can't really crystallize it because it's a dynamic system in lipids. And it's hard to do NMR on molecules that aren't bound together, that are loose, that are just sort of physisorb to each other. So we applied our method, and it's getting difficult now. We have the spectrum, and we have multiple peaks we looked at. So number one is the lipid head group vibrating that we always use. Then 2, 3, and 4 are 3 different peaks of vibrations of the tryptophan of the ring modes. Those 2 fused rings, indole ring is vibrating specific ways. So you can see there they are separated in SERS, and then the blue is Raman, and the black spectra are the DFT. And we can assign these peaks, and it's tricky, but we can. And as we do our analysis, we get high similarity at 4 places, but actually 2 of them have it going the wrong way, so we know those are wrong. And then the other 2 are actually mirror images of each other. So we actually only got one answer. And the answer we got is basically that ring is oriented in a way where the long axis of that ring is 53 degrees from the normal, which actually measures one other way you can do it. There's a way with polarization or polarized fluorescence, you can get this direction. So we got basically the right answer, 53 degrees. But we could also move the tryptophan in Z. And by looking at how, that's what this XY plot is, by looking at how the similarity maximized at Z, we could figure out where is the tryptophan sitting in terms of depth in the membrane. And that actually, there was no way to actually measure that. You can guess where it's going to go based on just chemical interactions, but this was the first actual, this is where tryptophan is sitting. And we're able to do it with this new method based on surface-enhanced Raman. So let's go on to conclusions. We've gone a long way through a lot of stuff. We should reward ourselves by being finished. So I don't want to drag you through anymore. So my conclusions are just, you can interpret SERS, right? So you can't interpret every peak, but if you use DFT, you can pick just the right peaks, and you can get structures out of SERS. So the 2 conclusions I showed you, one is that the CTAB is tilted at 27 degrees, and we could see lipid membranes as well, and we got the tryptophan orientation and position. The next slide is either acknowledgements or future directions. I forgot. Future directions is, yes, those conformational variations I told you about. If we want to get real, if we want to talk about real structures, we can't do the straight chain. We have to do the conformers. So here you can see how the map is changing with the conformers. And actually for CTAB, we've lost all our structural information if we get realistic about the conformers on that surfactant. So we got to publish a new paper that says, actually with conformers, we can't learn anything, but we'll still be able to learn things in membranes. But this is definitely where we're going to make things more real. It's just a lot more computational work. Finally, I must do acknowledgements. Of course, I've had many great grad students work on this project sort of over a decade. Probably who had to spend the most pain on it is James Matthews, the guy on the right. He and I got it started. And then Stephen Demers in more recent times has helped keep it going. But really everybody in my group works on this, including people that aren't pictured here. All these people made important contributions. And this was funded by the Welch Foundation and the NSF. So thanks for listening. And I'm happy to do questions in whatever format we can do questions.
Helps if I unmute myself. All of the things that were that your talk discussed, it was nice to see how physics can play in with the bio and the chemistry side of things as well. So it sort of blends the sciences together. Can you talk a little bit about how your research is more of an interdisciplinary project?
Yeah, so interdisciplinary, very important, and that definitely is the way the world of research is working now. So my lab is very along those lines because we, one way is just, this is a very weird combination of theory and experiment. Like usually you would sort of do this with a collaboration, but I found to really get exactly what I need and to have to just mess up and do the standard research progress of, okay, now we got to do it this way. Oh, we got to do it this way. Oh, we got to do it this way. It'd be hard to get a collaborator to do that. So that's why every step we go, we're like, okay, we just got to figure out how to do DFT. We just got to buy the package and start bugging tech support. So I'd say the main way this is interdisciplinary is actually between experiment and theory. Is it's sort of a pretty intense use of theory for one group to be doing. Not that I understand the theory better than a theorist, but just because the tool is available, I was able to really run with it. In terms of biology and physics, I mean, we're pretty typical for a biophysics group. There's lots of handling lipids and learning a little bit of the biochemical methods to keep them happy. And then a lot of the intense optics on the spectrometer side. But we're really more of a buy good commercial parts and then build something fairly custom. So we're not quite at the AMO level of building an optics setup, but we're also not using turnkey microscopes either. So somewhere in between. So it's a lot of optics, a lot of handling molecular systems or handling molecules, and then a lot of computational packages.
So I'll ask some of this on account of a couple of my students that that will watch this later or have watched it but don't like to ask questions. If students are looking into going into like your field of research or going into sort of this biophysics and nanomaterials type, what skills and or sort of academic background would you suggest that they pursue if they're looking, basically starting at sort of the community college level and working their way up?
Yes. So the field is very interdisciplinary. So that's good because in principle you could major in one of many things and move into nanoscience or into these sort of interdisciplinary fields. I mean, you can't go wrong with physics and chemistry. With those two, you would major in physics or chemistry, and then you could go to grad school in physics or chemistry, and those departments are definitely going to have people doing a bunch of different stuff. So if you like to tie it to fundamental things, physics and chemistry would probably make a lot of sense. If you're thinking a little more applied, or maybe if you want to keep your options a little more open after grad school or not, you could do a more applied discipline, and there material science or bioengineering or chemical, I mean, any of the engineerings basically, but material science and for biomedical applications, BioE is also a big one. And with an undergrad in those, you could, work for a company and directly get involved in this kind of thing, or you could go to grad school again in those kind of departments, which are also going to have these things. So it really just depends on it's more the fundamental or the more applied, but basically those four departments I would say would be my top choice if you want to go into stuff like this: physics, chemistry, material science, or BioE. And those are my general, there are more specific departments and many other options, but those would probably be a good four to look at kinds of majors.
And I know it's hard to put this into perspective as well, but we saw in your talk that sort of this field has evolved rapidly over a course of the last couple of decades. But can you sort of see where the field might be going in the next sort of in the next 5 to 10 years?
Yeah, that is hard to say because the field has become completely fractured into every area of science. It's sort of, what is nano now? So and I've been very sort of focused on what I'm doing the last couple of years. The best way would just be to pick a couple of, if you want to see where the field is headed and if it looks interesting to you, is I would say pick up, just browse the contents, even if you don't have access to the articles, just browse the glossy contents of ACS Nano and Nano Letters, and you'll see what I mean. The variety of things that people are doing is just, it's hard to even call it a discipline anymore. I mean, basically, those journals are coming almost applied journals, like applied science journals, because people are coming up with so many applications of nanoscience. There's still plenty of fundamental science happening, but it's almost all areas of science and technology are being impacted by the ability to control things at this scale. So I know that's not really an answer, and I'm sorry, but it's just sort of spreading its tendrils into everything is basically, I would say, the state of the field. There are, as always, there's a hot material every few years, right? I mean, there was graphene was really big, and now there's all kinds of other 2D materials is a big thing. So, you know, hot materials come and go, but the field is just sort of spreading everywhere, essentially.
Yep. Well, Dallas, do we have any questions from the chat in the various places, or we heard anything?
I have not seen any, gentlemen.
Okay. In that case, we can sort of wrap up here. Thank you very much, Dr. Hafner, for this talk, and very interesting results that I look forward to reading more about, and just an interesting field in general to look at. And I'm glad we got a chance to share it with the audience here, because we don't necessarily see a lot of sort of the biomedical applications that come out of this series. So I'm glad we got a chance to look at physics from that perspective this year.
Yeah, I was glad to be here. It was fun.
I do want to take a couple of minutes. I do want to take a couple of minutes to encourage any of the viewers to check out our physics department page at socc.com/physics, or socc.edu/physics rather, or check our department Facebook page, because we have a number of we have a number of speaker events within the series. This is the first of, I believe, 6 this year that we'll have over the course of the year, and we have a number of student opportunities with NASA funding and student invention competitions that'll be ongoing over the course of the next couple of months. So stay tuned to our social media and to our website to find out more information about that. And we are always happy to have and visiting scientists like Dr. Hafner that support our series and our attempts to get cutting edge science out to the coastal Oregon community. So I thank you guys for attending, and I hope to see you next time.